those keeping track, 2 more talks, and you're done. Well, I guess there's a lot of congratulations after that. There's at least 2 more scientific presentations. So I'm going to completely switch gears and talk about a crop that's not been discussed at all, I think, and that's mostly soybean. And I'm going to talk about bottlenecks and sort of a lack of genetic variation that exists in soybean breeding populations, and specifically in the U.S., although it's true in other countries as well where there's active soybean breeding programs. And I think most people are familiar with bottlenecks, and of course, if you've done any driving or been out in a car anywhere in major cities in India, you're also familiar with bottlenecks. So just to quickly illustrate, this is my only slide that's not soybean, bottlenecks. This is some sequencing that we've done in common bean, Phaseolus vulgaris, across the range of distributions, so from North Mexico all the way down into the Andes in South America. And you're probably aware that common beans had 2 independent domestication events, one up in what we call Mesoamerica, another one that occurred down in South America that we call the Andean domestication event. And so we did a range of sequencing across this distribution, looking at land races, looking at wild relatives, looking at early domesticates, and looking at the wilds, both from the Mesoamerican and from the Andean populations. We're able to model what the historical demography of these populations look like. And there's likely an ancestral population that existed somewhere in Mesoamerica, and that population's remained relatively unchanged as far as introduction of any bottlenecks or changes in genetic variation. But there's a migration of a small population down into the Andes, and that experienced a bottleneck that you see here, which lasted for about 60 to 70,000 years. And in fact, the effective population size during this bottleneck was about 3,600 individuals based on modeling of data from the sequencing. And so this just sort of illustrates in real terms what a bottleneck can look like. And of course, this is not the domestication bottleneck. This is just a bottleneck due to migration from the north to the south. And we can also estimate what the exchange of genes were between the north and south. So that's just sort of an illustrative example of what a bottleneck can look like. And now I want to switch to the genus Glycine. So this is the genus that contains soybean, Glycine max. That's the cultivated form of glycine. It's a domesticated form of what we call Glycine-soja, and these are often referred to as just a species complex, and Glycine max is just a cultivated form of Glycine-soja. So domestication probably occurred somewhere in northeast China along the Yellow River, and this is sort of the range of distribution of Glycine-soja, Glycine max. The rest of the genus, these are all perennials, and these are almost exclusively found in Australia, although there are a few found up here in Asia as well, but there's about 26 species that have been characterized thus far, found exclusively, almost exclusively in Australia. And so you can see the changes between the domesticated form of soybean versus the undomesticated forms. You have smaller seeds, darker seeds, you have shattering in the Glycine-soja. Very typical things when you look at almost any plant that's gone through domestication. So if you just sort of think about what's happened with soybean over the past 10,000 years or so, of course, domestication occurred somewhere in China in the last 6 to 9,000 years. That's imposed a bottleneck of some sort. There's been adaptation that occurred both before and after domestication. So we know soybean has a very, very strong response to day length. So there's been adaptation to longitudinal regimes that occurred both before and after domestication. If you think about what happened as far as the U.S., the first soybeans were brought to the U.S. in 1765, coincidentally to Georgia, where I'm from. So there's a bottleneck in the number of introductions that came to the U.S. at that point. And I think the most interesting bottleneck, and this is based on modeling or analysis of breeding programs in the U.S., almost all the breeding germplasm in the U.S. traces back to 12 founder lines. And that's an incredible bottleneck if you think about the genetic variation that's present in modern breeding programs in the U.S. And that's breeding programs in companies, it's public breeding programs, all trace back to about 12 founder lines. And you sort of diagram what this all looks like, and there's been a very, very strong bottleneck in the last 50, 60, 80 years in the U.S., at least with these founder lines. And you would see a similar picture in China, you'd see a similar picture in Brazil where there's major or active breeding programs. And so one of the things that has been of interest to the soybean research community, to the various companies, and to the growers, is how do we capture more variation within breeding lines that we can begin to combat some of the things that we know are coming down the line as far as climate change, differences and shifts in pathogens and pests that attack soybean, and to keep the increase in yield potential. So there's all sorts of, well, there's several sources of variation that you can think about. There's standing variation that exists in our breeding populations, and I think if you talk to the breeders, what's mostly happening now is they're just shuffling haplotypes. They've accumulated superior alleles in phase, and they're shuffling haplotypes within populations, and they continue to make gains in yield. There's de novo or induced mutation. Actually, induced mutation, EMS mutation, has been used to create mutations in genes and oil biosynthesis. We've had changes in oil composition due to induced changes. And then there's the potential of the wild relatives, and that's what I want to talk about. The wild relatives exist for the genus Glycine. And then I'm going to talk a little bit about epigenetic variation that exists within cultivated and also wild soybean. So I'm going to talk first about the perennials, these things that exist down mostly in Australia, then about the annuals, Glycine-soja, and then some of the land races and Glycine-max, and then the epigenetic variations. So the genus, if you look at where they're distributed across Australia, they're found everywhere, from salty areas along the ocean to the outback, to the mountains, to very dry, to very moist areas. So there's a lot of variation in adaptation within these species. This is just a subset of the species showing their geographic distribution. And we know from phenotyping and looking at these things that they have all sorts of resistance to things that beset production of soybean. So that some of these things have resistance to soybean cyst nematode. You grow them in infected soils. There's no infection. So various other pathogens that affect or attack soybean. And so with a group of collaborators, we made a variety of genomic resources, made back libraries, anchored these back to the genome sequence that you could traverse. There's about 5 to 8 million years of evolution within the genus Glycine. So if there's a region that you're interested in Glycine-max, the domesticated form, you could traverse 5 to 8 million years and look at what the orthologous genes and potentially map and clone genes from these wild relatives. We did LD studies in wild populations, so you could then associate traits with genomic regions and various other things. But at the end of the day, it's actually, we should have known this going in. There have been decades of work of people trying to make crosses with the perennial glycines. Multiple groups, almost entire careers, they've spent trying to make crosses between Glycine-max and these perennial glycines. And out of all that work, there's been maybe 2 or 3 successful crosses with 1 or 2 of these wild perennial glycines. So it's not very tractable for bringing in through crossing. And this is in contrast to other species I've worked on, like rice, where you can make crosses going out 5, 8, 10 million years. You may have to use embryo rescue, but you can get fertile progeny. But decades of work, there's only a handful of crosses. And in fact, most of these crosses result only in chromosome addition lines. There's almost no recombination between the chromosomes from the perennials and the annual chromosomes. And of course, the non-agronomic performance of these, which is true of a lot of different diversities, so it's not so much a factor. And there's also recurrent polyploidy. So there's 2 rounds of polyploidy that exist in this branch, but there's additional polyploidies that have occurred in the last 50,000 years with these things that makes it even more complicated to work with these either at the genetic level, genomic level, or crossing. So really, the only way, in a tractable sense, to begin to move genes from these perennials is through cisgenesis, and that's cloning the genes and moving them through transgenesis. So in my lab, we decided to focus more on this branch here, which is the Glycine-soja, Glycine-max complex. Of course, max is the domesticated form, which also has 5 to 8 million years of evolution. It's the basal branch in the Glycine genus. But these things are crossable. So you can cross the wild Glycine-sojas with the Glycine-max. You can get progeny. You can get recombination between the chromosomes, and you begin to move genes in. And there's a lot of genetic variation in this branch that has not been tapped. And so this is where we now focus most of our work, is in this branch here. So as I said before, Glycine-max it's really just a domesticated form of Glycine-soja. Domestication, if you look at nucleotide diversity, this is work from Perry Cregan's lab, reduced variation by about 1/2 going from soja to max. And that number, both those numbers are really small if you look at nucleotide diversity, say in maize, between inbreds. So there's even going to the domesticated form, there's not a huge amount of genetic variation compared to, say, maize or wheat even. So there's about 5 to 8 million years of evolution within that lineage, within that branch. We know there's many potentially useful traits. Many of the ones I showed you before, there's also resistance for those various pests and pathogens within Glycine-soja. Unfortunately, we don't know very much about the genetic and genomic variation in Glycine-soja, and that's because, obviously, most of the genetic activity in the last 50, 70 years has been on the domesticated form. So a number of years ago with Suka Ali, who I think has already left, we published a single genome for Glycine-soja. And basically, it was not a genome assembly as much as it was mapping Glycine-soja reads back to the reference genome. So working with a group in China at the Chinese Academy of Agricultural Sciences, based on some of their work where they had done genotyping across a large collection of Glycine-sojas, we selected 7 accessions based on structure, so population structure, and also geography to represent where the distribution of Glycine-soja. So from the south of China to the northeast, Russia, Korea, and Japan as well. And we did Pan-Genome, or we're creating a Pan-Genome. So we did de novo genome assemblies for each one of these 7 accessions representing the population structure and the geographic diversity of Glycine-soja. And we sequenced each of these to about 100x coverage, did genome assemblies. These are, so those are really interested in genome assemblies. We can talk about this later. This could be vastly improved if we did 10, 20, 40 kb jumping libraries to get more contiguity in scaffolds and contigs, but this is, the contig and scaffold sizes we have now range from about 8 to about 30 kilobases in contigs, and the scaffolds max out about 60 kilobases. As you might expect, the length of the contigs and scaffolds is directly correlated with the heterozygosity in the original plant. So the more heterozygous it was, the smaller the contigs and scaffolds were. But this creates what we call the Pan-Genome. And there was a discussion, I guess, 2 days ago about how you sample things, especially when you have presence-absence variations. So you have a reference genome, you have things that are not present in that, but they're present in another sample. How do you begin to build these into some display or some analysis or investigation of diversity? One way to do that is to build a Pan-Genome that represents a broad range of diversity within a genus or within a species. And that's what we've done here with these 7 accessions. And so we're now going back in and resequencing within these geographic distributions and mapping back to the Pan-Genome or to the genome representative of that distribution. So I mentioned before, the Glycine-soja glycine branch is about 5 to 8 million years. It's a basal branch within the genus Glycine. And so here's the 7 accessions. Here's Glycine max. Of course, when you compare it to other cultivated or other crop legumes like common bean, everything collapses down. When you compare amongst themselves, it turns out that these Glycine-sojas that we're sampling diverged about 600,000 years ago. And they diverged from Glycine max about 0.8 million years ago, about 800,000 years ago. You can take these estimates with a large grain of salt. And these are the deepest things that we have. So there's a lot of things that don't exist anymore from that earlier 5 million years or so, and this is what's left. But we've also not sampled much that would be out here, except for the land races. So we're not sure exactly from where. We suspect it's in the northeast where domestication occurred. We have to do more sampling to find Glycine-soja that would be more closely related to the domesticated form of Glycine max. So quick description of the Pan-Genome. So it's about, gene count's about 60,000 genes within the Pan-Genome. Of course, because the contigs are so small, this is inflated because you have genes that are broken across contigs. Soybean has about 55,000 genes within the reference genome. The core genome, that's conserved amongst all 7 accessions, forms about 86% of this almost 1 gigabase. About 14% is dispensable. If you look at what's lineage specific, it's actually about 6% of that 14%. So there's about 6% that we found only in one genome, one of the 7 genomes, versus shared among some subset of those 7 genomes. But if you look at the gene set, so there's about 60,000 genes. About half of those are conserved amongst those 7 genomes. That means the other half is dispensable among those 7 genomes. And if you start thinking about soybean, which has about 70+% duplicated genes, you can have a lot of shuffling and gain loss of genes without losing a function because you can have a duplicate copy elsewhere in the genome. So maybe it makes sense that half of it can be dispensable. But in any one genome, you have between 1,000 and 3,000 genes that are lineage specific. That means they're not found in any of the other 7 Glycine-soja genomes or even in Glycine max. So some of these are probably not real genes, and we can talk later about that. They're annotated as genes, but things should be annotated as genes, but they're not really genes. But a subset of those are genes based on RNA-seq and homology to other genomes. So looking at that 60,000 genes, we're looking at just doing a test for selection within those lineages. So this is a maximum likelihood based on phylogeny approach. I really can't explain it any more than that. It's been explained to me several times, and I'm still not quite sure I grasp it all. But with any one of these lineages, about 1 to 2,000 genes that appear to be showing under positive selection. It might be involved in some adaptation to growing in that particular region. So in any one of these glycine sojas, about 1 to 2,000 genes that appear to be under adaptive or positive selection. If you contrast it to glycine max, doing the same comparison is about 5,000. So there's many more genes that show positive selection or adaptive selection within glycine max versus these glycine sojas. But it'd be interesting to know what the function of these genes under adaptive selection are and what use they might be in moving and testing in adapted cultivated soybean. So there's a lot more. I just want to quickly summarize this part. So we have a pan-genome for the wild glycine soja. This is not a pan-genome for domesticated. That's a separate project. This is a pan-genome trying to look at the diversity and sort of variation within the wild progenitor glycine soja. About 60,000 genes, about 86% of the genome is conserved among those 7 accessions. There's lots of variation, which I didn't talk too much about, in retention of duplicated genes, showing lineage-dependent retention of paralogs. And we're also seeing this if we do resequencing across the cultivated forms, across accessions within glycine max. You also see lineage-dependent retention of duplicated genes or paralogs. Talked about the genes under positive selection, a few thousand in the glycine sojas versus 2 or 3 times more in glycine max. And I think it's interesting to start looking at these genes, especially those that might be in the dispensable genome or those under positive selection, begin testing these in cultivated soybean, see if there's some value in these things and moving these forward into soybean. They may have been under positive selection for something in response to some environmental cue or some environmental shift within that region that they're grown in that could be valuable, at least for domesticated soybean. So I'm going to wrap up a little bit. So another source of variation is epigenetic variation. And of course, when you go to genetic conferences, if they ask a question you don't understand, the response is, it must be epigenetic. So I'll talk a little bit about epigenetic variation. So epigenetic variation is just variation that there's not a causal DNA change or a change in sequence or deletion or insertion or something in the nucleotides, but has more to do with the way the DNA is packaged. So the methylation of the DNA, the way it's packaged into histones, the modifications of the histones are associated with the packaging, all part of this complex that we call epigenetic variation. The most easy to interrogate is the cytosine methylation or DNA methylation, because you can create specialized libraries, sequence those, and you can look at the DNA methylation. And so just a quick diagram of what this might look like. We're looking at cytosine methylation. You can have cytosine methylation in symmetric sites that we call CG. We're showing that in red. And then you have asymmetric methylation in CHG and CHH, in blue and green. And typically, when a gene is expressed, you have CG methylation. You have CG, we call gene body methylation, CG methylation. And very often there's a correlation. The increased CG methylations, there's a correlation with increased expression of that gene. So you can have that type of methylation within the gene, and you still have the transcription. When you start to see CHH and CHG methylation, this asymmetric methylation, that's when you begin to see genes not being transcribed. So this is an example. This is actually a transposon. You have CG and CHH and CHG methylation. It's also very often correlated with accumulation of 24 small interfering RNAs via a pathway that's called the RNA-dependent DNA methylation pathway. And that results in the gene, or in this case a transposon, not being expressed. So this is the RDDM silencing pathway. You have a gene, you get a transcript. RNA-dependent RNA polymerase creates a double-stranded, goes to a diester, creates 24 nucleotides. This is all associated with the argonaute that comes back and establishes a methyl fingerprint at that site that would include the CHG and CHH methylation and result in that region being shut off for transcription. So here's what it looks like in soybean. So this is actually an annotated transposon in orange. So I'm going to show you a couple of these things, so I just want to go through this quickly. So in orange is an annotated transposon. And then in these various tracks here, we have CG in red, we have CHG methylation in blue, CHH methylation in green. The 24 nucleotide RNAs are in yellow, and RNA-seq data is in this track here, which is shown in red. So this is a transposon. This is very, if you're looking for a locus that shows all the hallmarks of RDDM methylation, it has the 24 nucleotides, it has a CHH and CHG methylation, and has no transcription. Very typical of an RDDM target locus. This is right next to it is an active gene. So you have CG body methylation. You don't have any CHH or CHG methylation. Very small numbers almost insignificant numbers of 24 nucleotide, and you have transcription as shown by the red mountains on top of the exons. Here's another transcribed gene that actually has a TE within an intron that's showing the RDDM pathway, but it's not affecting the transcription of the gene that's embedded within. So one of the things we're interested in, we've had a long interest in sort of the evolution and polyploid that's occurred within various crop plants, but especially soybean. So soybean has 70+% of its genes are present in multiple copies. This is due to 2 rounds of whole genome duplication or polyploiding. And we know that within any one tissue, about 40 to 50% of the genes show divergence of transcription. That is, one is expressed, the paralog is either not expressed or the expression is greatly attenuated, so it's much less expressed than the other copy. And so one of the things we've wondered about is what sets up this pattern of differential transcription within tissues between paralogs. And one of the things, this catch-all phrase would be epigenetic, must be epigenetic regulated. And that's one of the things we're investigating. And so one of the ways that a gene can be turned off, and there are classical examples of this, is if you have a transposon either in or near the gene, and you can have spreading of this asymmetric methylation, the CHH and CH gene of the gene, that would turn that gene off. And so there's examples like FWA and BNS, where there's a transposon that actually causes the silencing of the gene due to spreading of methylation marks. And so we wanted to explore within the soybean genome, and particularly looking at these duplicated genes, what the genomic context, what transposons might have an effect on the silencing of paralogs within certain tissues. And we quickly ran into a problem, and we should have known this going in, because my lab's worked on transposons for a number of years, and every genome we've worked on, and every genome that's out there, even the best annotated genome you can think of, like Arabidopsis, there are still transposons that are annotated as genes, even though they're transposons. And so we should have, this should have been the forefront of our mind going into this. But as we began to look for genes that showed hallmarks of the CH, this asymmetric methylation, shown in blue and green here, we were also finding lots of these that had 24 nucleotides, which is not correlated with the spreading of the DNA or the spreading of the methylation mark. And so the question is this an actual gene, or is this a transposon that's been annotated as a gene? And it turns out in this particular example, it is a transposon that's been annotated as a gene. But we wanted to come up with a way to go through the genome and better annotate these cryptic transposons that are annotated as genes to remove them from our analyses. And so one method we came up to do that is to take the 24 nucleotides themselves as a hallmark of transposons, or these repetitive elements that are silenced by the RDDM pathway within the genome. And so we used a window approach to map the 24 nucleotides across the genome. Of course, transposons and things that are transposon-like are silenced by this pathway, so they have 24 nucleotides that map within them. Genes typically do not. Pull these things out, cluster them, and build families based on that. And here's just what the pipeline looks like. So you map the 24 nucleotides, extract fastest sequences, do an all-by-all blast to find families, go back and extract the flanking sequence, and you define the exact borders. And then we use a tool to annotate transposon-like motifs, and then add this back to the library to use to match the genome. And here's what the results look like. So the original soybean transposon database had this 75,000 elements, maps about 48% of the genome. If we add this annotation, we have about 100,000 elements, and we mask about 60% of the genome. And so going back to that example I showed you before, we now have this track in green. These are the newly annotated transposons based on this mapping back to 24 nucleotides, and then the annotation at the end to ensure that they're actually transposons. And about 2.5%, 3% of the genes within the soybean genome are transposons annotated as genes. There's probably a greater number. That's the number we're able to extract using this approach. So now we can go back in and in a more refined way begin to look at how methylation affects paralogs, or just genes in general within soybean. So here we have an example of a typical normal gene. We have the gene annotation here. We have transcription. We don't have CHH, CHG methylation, just some CG body methylation. Here's a TE gene. This is the one I showed you before. So it's annotated as a gene that has all the hallmarks of an RDDM-targeted transposon sequence in the genome, and it's now annotated as a TE. And then these are the things that we're really looking for. Things that are annotated as genes. This happens to be a single exon gene. Has the asymmetric methylation in the green and blue here, but doesn't have a 24 nucleotides that would target it, or would say that it's a TE annotated as a gene. And of those, there's about 3,000 in the genome. So now we have a set of genes that show asymmetric methylation, that have attenuated expression patterns. And now we can begin to look at these to see if these are due to spreading effects or other some contextual environment, and what the end result of these might be within the soybean genome. So we call these methylated genes. I mean, genes are methylated anyway at CG, but these are methylated both at CG and CHH and CHG and CHH. So I'm going to refer to them as methylated genes. So they're enriched in pericentromeric regions. This makes a lot of sense. Pericentromeric regions have a lot of TEs. There's a lot of RDDM targeted loci within pericentromeric regions. So if there is a spreading of this type of methylation to genes, the genes that are in that region, it makes sense that they'd be enriched in that fraction. So 25% of the total genes are in pericentromeres, but 70% of our methylated genes are in those regions. I'm not going to go into this, but based on RNA-seq data and also on looking at the nucleotide changes, it looks like they're enriched for things that are being targeted for pseudogenization. They're also enriched for being duplicated. So roughly 70%, slightly less than 70% of the genes in soybean are duplicated. Of these, there's about 90% of them are duplicated. And this also makes sense. So if you're going to shut something off by this pathway, having another copy in the genome that can be expressed and recover the function does make it makes some logical sense. So this is just an example of one set of paralogs we call paralog A. This is on chromosome 2, paralog B on chromosome 18. This gene's in this orientation, this gene is in this orientation. They both have transposons in their introns. Both of the transposons targeted by RDDM pathways. You have the small nucleotides, you have the asymmetric methylation. Same thing here. What's different is this gene also has asymmetric methylation over its entire length of the gene. There's also enriched the number of 24 nucleotides that map to this transposon that's an intron are much higher than this one here. And there's a weak correlation. So when we do find these types of genes, so this one's expressed at low levels. You can see low levels of expression. It doesn't have the asymmetric methylation on the exons or introns across the rest of the gene, except for where the TE is. So this one's expressed. This one's not expressed. This appears to be targeted by the RDDM spreading, perhaps. And the weak correlation is that when we do find these things where there's TEs in both genes or upstream or downstream both genes, the one that has what appears to be a spreading effect, the number of small nucleotides that map to the TE tend to be greater than the one that there's expression. There's not the spreading of the methylation. It's a very weak correlation. So if we sum that data across all those sets of genes, so here we have, just think about an evolutionary sense, you have some gene becomes duplicated. One of the paralogs now shows this symmetric and asymmetric methylation, potentially due to spreading from TEs. This is whisker plots of DNA methylation, CG, and then the two asymmetric. This is the paralogs, have both lower CG, but much, much, much lower asymmetric methylation. And if you look at the transcription across the entire data set of those genes, you can see that the genes that are methylated at all three contexts have almost no transcription versus the paralogs don't show that. There is variation in expression. They are expressed at some level. So framing this in a broader evolutionary context, we've also produced similar data sets for phaseolus and Glycine-soja. And so we can look at the methylation of the paralogs within Glycine-max. We can compare what's going on in Glycine-sojas and also the orthologs in phaseolus. So phaseolus does not have the most recent duplication that's found in glycine. And so this is the same data that I showed you before. So the three methylation contexts in soybean. Here's the orthologs in common bean, which don't show any of the CH or CHG methylation, but do have some level of CG methylation, which is typical in genes anyway. And looking at the expression patterns based on RNA-seq data, they're also expressed, which suggests that if wherever this mark was set, it was set after the divergence of phaseolus and Glycine-max, and probably after the duplication event that occurred most recently within the glycine lineage. And I'm not going to show the data, but if we start looking at these across accessions with Glycine-max and Glycine-soja, you see some sorting of these methylation marks between paralogs. It's not always the same paralog that shows the mark, although the ones headed for pseudogenization are consistently marked. So, almost final slide here. So if you think about the fate of these genes, you have some genes, it's duplicated, so the most recently duplicated genes in soybean about 8 million years ago. One of the genes gets shut off via this pathway, either spreading or some other pathway where you get the CHH and CHG methylation as well as CG methylation. And so I talked a little bit about genes that might be headed for pseudogenization, and that's some maybe 20% of the genes that appear to be headed towards pseudogenization. What I didn't present is some of these genes that show this mark. If we look at RNA-seq data, expression data from endosperm or from tissues where there's a meristematic reprogramming, they're turned back on. So the methylation mark is removed, and these genes are shown expression. And so there's actually data coming from Arabidopsis and some other work where genes that show typical RDDM type of methylation, or the 3-cytosine methylation context, are often released in stress, under stress, potentially when there's a pathogen infection, or in shifts in meristematic identity or meristematic reprogramming. And then I mentioned, but I didn't show you the data, so if we look at these particular genes across the Glycine-soja Pan-Genome, or even within Glycine Max, looking at different accessions, some of them, there's switching of off and on, so it is reversible. It's not set completely. So there is some assortment and shuffling of these things. And so what we don't know, there's a lot of work to go on, is what is the standing level of epigenetic variation, what role does this play in phenotypic variation, and how has this been selected on during breeding? And that's something we're working on currently. So I think I've said all that. I'm done. So I want to thank my collaborators. So the Pan-Genome was done in collaboration with the group in the Chinese Academy of Agricultural Sciences. The common bean stuff, which is one slide, was all those people. And soybean methylome is work in my lab from some very talented postdocs here, and these are our funding agencies. We should always close with something. This is the Olympics, right? So there's this great quote from the Dutch skating team coach today, Fred, telling the Americans why we didn't win any skating medals. It's because football sucks. So anyway. Right, so we haven't done the pump. We're doing anthers right now. Right, no, I mean we thought about that. And actually Bob Schmitz, who's one of the authors in that, is part of our project now. So, and that's one of the things. And the pollen's very interesting because everything's released. So you're asking about the spreading within homeologs. So, right. So the problem, there's a couple things. So in soybeans, it's been so fractionated, we don't have complete chromosome level homology or homeology or whatever. And we've not yet looked within, so we have blocks of Cincinnati. We've not yet looked in those, but so some of our RNA-seq data, we're looking at differential expression of duplicated genes. Over entire blocks, you can see that where one copy is more highly expressed versus the other copy. So we're planning to integrate that, but we haven't done that yet. It's a good question. It's a logical extension.
It is quick. Really enjoyed the talk. Your pan-genome analysis, was it 7 or 9 individuals you resequenced?
Pardon?
For the pan-genome analysis, you resequenced and reassembled, was it 7 or 9 individuals? So do you think you've captured the vast majority of extra genes there, or do you think it's worthwhile going out and sequencing some more individuals? And if you do, how many do you think you need to be able to capture 99% of the genes that are out there?
So I didn't present it, but we've actually done analysis of, with each genome that you add, how much more do you capture? And it began to asymptote around 6 or 7, but we need to sample more to make sure the asymptote was real. We're in the progress of doing resequencing, not additional genomes, across those sets of geographic ranges. And once we do that, then we'll know, how much more things aren't mapping and assemble independently. But I think we're close.